Hello everyone, and thank you for joining me for this first Nanolive webinar of 2021. For those of you who don't know me yet, my name's Dr. Emma Gibbin-Lameira, the Communications Specialist here at Nanolive. Today's talk will be focused on quantifying dynamic changes in cell metabolism. I'll begin with a short introduction to the topic before introducing you to our technology. We'll then move on to the main part of the webinar, which will be split into two parts. The first part will feature the real-time response of mitochondria to respiratory inhibition, and the second part will focus on quantifying lipid droplet dynamics. As usual, I'll finish with some conclusions and discussion points before moving on to a short question and answer session. You can ask questions by typing them in the box at the bottom of your screen. It's Nanolive's policy to moderate discussions during live meetings, and so your questions will be sent to us by email, and a member of our team will select 3, pass them to me so that I can answer them at the end of the event. Okay, so let's get started. I'm sure you all recognise this image of a typical eukaryotic cell containing compartmentalised organelles surrounded by cell cytoplasm. This is a staple in any biology textbook, and so students could be forgiven for thinking that organelles maintain a nice orderly distance from one another. In fact, this is far from the case. Mitochondria form dynamic physical connections with other membranous compartments, including the endoplasmic reticulum, lipid droplets, Golgi apparatus, lysosomes, melanosomes, and peroxisomes. The relationship between mitochondria and lipid droplets is particularly important as this forms the foundation of cell metabolism. Mitochondrial activity essentially controls whether lipid droplet reserves are expanded via lipogenesis and triglyceride synthesis or consumed via mitochondrial fat oxidation. Disruptions to mitochondrial dynamics therefore have serious health implications. Mitochondrial dysfunction, the overproduction of reactive oxygen species and the reduction of ATP levels, has been linked to type 2 diabetes, neurodegenerative diseases, and some cancers, whilst dysfunction in metabolic excess can lead to non-alcoholic fatty liver disease and insulin resistance. The commonality of mitochondria to so many different pathologies makes them attractive therapeutic target for drugs. So how do we currently image changes in cell metabolism, and what does Nanolive have to offer? Conventional high-throughput live cell imaging approaches can be divided into 2 parts. That's label-free versus fluorescence-based imaging. Label-free imaging, such as phase contrast microscopy, as you can see on the left of your screen, allows cells to be observed at high frequency for unlimited periods of time, but lacks resolution, contrast, and dimensionality, making it impossible to distinguish details at the cellular organelle level, and easy to miss critical or transient effects. In contrast, fluorescence-based techniques suffer from phototoxicity and photobleaching, which perturb cell morphology and impose trade-offs between the duration of imaging and the frequency that you can do during your experiment. Again, this makes it easy to miss important events during the course of the experiment. Multiplexed imaging, at least at the population level, is not currently possible, making it impossible to measure multiple metrics simultaneously. In contrast, Nanolive imaging offers a unique, label-free solution to imaging single cells. Our microscopes measure a cell's refractive index via holotomography, an approach that generates no phototoxicity and no photobleaching. This makes it possible to capture the real response of cells to drugs at high frequencies and over unlimited periods of time. Our high precision segmentations that have been playing on your screen as I speak allow to quantify multiple cell metrics simultaneously. Here we show confluence as an example, but the other metrics are listed on the screen. And because different organelles have different refractive indexes, we can visualize the simultaneous response and interactions of organelles with subcellular resolution. Multiplexed imaging offers a unique opportunity to study dynamic changes in the nucleus, in the mitochondria, particularly fission and fusion events in the mitochondria. It allows us to see the mobility and the growth or depletion of lipid droplet reserves, observe the recycling activity of lysosomes, and visualize the formation of endocytic vacuoles. And all of this we can see at the same time as monitoring changes in cell behavior via the membrane protrusions. So let me show you now how Nanolive cell imaging can be used to visualize changes in mitochondrial morphology following perturbation. In this example, we exposed 3T3-derived preadipocyte cells to 100 micromolar of antimycin A, which is an inhibitor of cellular respiration. Images were taken once every 6 seconds for 15 minutes using our 3D Cell Explorer fluid microscope. And when I start the video here, the thing that you need to watch out for is a change in mitochondrial shape. So at first the mitochondria are long and tubular, but very quickly the addition of the inhibitor causes the mitochondria to become swollen and ring-shaped. This is likely due to a loss of membrane potential, which triggers the collapse of structural support mechanisms and renders the mitochondria dysfunctional. We can monitor the moment mitochondrial dysfunction occurs using a cell metric called granularity. Granularity is a texture measurement that describes how the refractive index signal is distributed throughout the cell. So this is always relative to a certain scale. Here we use 5 pixels. A change in granularity, here we'll see in a minute that it increases in this cell, indicates that there's been a redistribution of organelles within the cell. And this measurement this makes this measurement particularly interesting for holotomographic images, which are texturally more complex than fluorescent images. In some examples, we've observed subtle changes in granularity precede apoptosis, such as in this preadipocyte cell, which was exposed to mitotracker and fluorescence excitation and imaged at the same rate as before. In this video, we see that the cell dies after just 28 minutes. This highlights the negative impacts that phototoxicity has on the mitochondrial network and on cell health in general. However, in other examples, such as in this population of cells that were exposed to 25 micromolar of antimycin A, and imaged using the 10x10 grid scan mode on our automated microscope, the CXA, we observe a change and subsequent recovery in granularity. This suggests that in certain conditions, cells initially react to treatment, but then are able to recover. Maybe that they rearrange, for example, their cellular composition in response to a certain drug. So understanding the link between cell health and granularity requires further investigation, but looks like it could be a very promising avenue for research in the future. Now let's move on to part 2 of the webinar, lipid droplet analyses. In this part, I would just like to take a minute to introduce a different type of technique which is specific for measuring lipid droplet analyses, and that is Raman spectroscopy, which provides a label-free alternative to fluorescence-based imaging, but also suffers its own limitations. So this technique is costly It's extremely complex and suffers constraints from signal specificity and temporal resolution. As I showed earlier in the webinar, our technology overcomes all of these limitations. And now, even better, our analysis of lipid droplets is fully quantitative. So in this part of the webinar, I will reuse part of the dataset that I showed in my last webinar on single cell characterization. The dataset features the growth of a population of 3T3-derived preadipocyte cells over 30 hours, during which confluence increases by 26%. The close-up that is now playing on your screen shows the high precision of our lipid droplet segmentations. The most basic type of analysis that we can do at this population level is to count the number of lipid droplets at each time point and see how that evolves throughout the experiment. This is plotted in the graph on the left of your screen. Lipid droplet count is clearly stable for the first 12 hours, but then starts to increase over the next 24. A similar trend is observed in the graph on the right, which shows the lipid droplet dry mass content over time. So as we expect, the two show exactly the same pattern. We can take the analysis one step further if we calculate the average number of lipid droplets per cell. We see an initial reduction and then a subsequent increase in the number of lipid droplets per cell. It's possible that this could be linked to cell senescence, although further experiments would be needed to determine that. There are several studies that have reported that lipid droplet accumulation occurs in older cells in the population. Alternatively, this could simply reflect a change in energy homeostasis as confluency increases. Correlating the number of lipid droplets with other parameters, other morphological traits may provide a clue. So here I've plotted average lipid droplet count against cell area, compactness, dry mass, granularity, extent, and perimeter. We can see from these scatter plots that three traits, perimeter, extent, and granularity, are positively correlated with lipid droplet count. Two traits, compactness and dry mass, are negatively correlated with lipid droplet count, while no correlation exists between cell area and lipid droplet count. It's important to remember that with correlations, this does not equal causality. So this simply allows us to describe what an average cell looks like. Our data from this experiment then suggests that cells that contain more lipid droplets have a larger perimeter, a larger extent, and a higher granularity, but also tend to be less compact and contain less dry mass. This last finding is particularly interesting. There are two possible explanations for this. So the first is that individual lipid droplet may be smaller in cells that contain more of them. Or the second possibility is that cells that contain less lipid droplets may have more cytosolic or nuclear dry mass. We can test the first hypothesis by plotting whether individual lipid droplet dry mass content changes over time. So I'll let the graph play for a minute. But what you will see is that it fluctuates slightly, but does not change drastically over time. If we look at a correlation between what an average lipid droplet looks like and how much it weighs, so its dry mass content, we can see that there is a weak negative correlation between the two. This suggests that individual lipid droplets tend to be smaller if a cell contains more of them. So it favours that first hypothesis that I put forward on the last slide. So the suggestions that I've made here today are simply that. They're just examples to show you of the type of analyses that is now possible using Nanolive imaging. If you're looking for further inspiration on how you could use Nanolive imaging, then I would point you to this excellent article published last year in PLOS Biology by the Dean of the Life Sciences, department at EPFL, Professor Gizu van der Guut and colleagues. So in this article, oleic acid is used to trigger lipid droplet accumulation in HeLa cells. The authors then quantify the number of lipid droplets and quantify how their dry mass evolves over time. After 3 hours of exposure to oleic acid, lipid droplet count increases 16-fold, while individual lipid droplet dry mass content increases 6 times, is 6 times higher at the end of the experiment than at the start. So this, for example, would be a good technique for looking at lipid droplet formation and calculating rates of how they grow over time. So I invite you to check out that publication if you have the time. So plenty of food for thought for today. That's all I have time for now. So if we move on to some conclusions. In today's webinar, I've introduced the concept of granularity as a potential label-free marker of cell or possibly mitochondrial health. I've showcased the possibilities for label-free quantitative lipid droplet analysis at 3 levels. So at the population level, at the single cell level, and at the individual lipid droplet level. And we are very confident here that the new analysis tools that I've described today really permit us to access an uncharted field of biological dynamics. But this is just a small fraction of what is possible using our technology. So if you do want to know anything further about the image analysis tools that I've explained today or that we provide for our clients, then please don't hesitate to contact me in the future. Now let's move on to the question and answer session. So, just 2 seconds. Question 1 is, have you examined mitochondrial morphology in cell types other than pre-adipocyte cells? And if so, have you noticed differences in morphology between cell types? So yes, we've imaged many different cell types, all of which you can find on our website. Off the top of my head, I know we've imaged mesenchymal stem cells, neurons, HeLa cells, various different types of cancer cells, and we can see mitochondria in high resolution in all of them. The mitochondrial morphology differs quite a lot between different cell types. So in some cell types they're long and elongated, in others they're more spotty and discrete. However, we have to be aware of the fact that the mitochondrial morphology is directly related to their metabolic activity during that experiment. So that can also change across time within a cell type as well. Okay. Question 2 is, have you tried to observe differences between starved and non-starved cells? And can you see contact points between mitochondria and lipid droplets? So on many occasions we've looked at starved and versus non-starved cells in the past. All of the videos that I've shown today though are from non-starved cells. And so the differences that you expect in morphology are, well, it's what you would expect really. So starved cells have less lipid droplet reserves because they're used up. They normally have longer mitochondria that are thinner because they're more metabolically active. And the second part of the question was about contact points. So yes, the resolution of our microscopes is, you can clearly see the contact points between lipid droplets and mitochondria, which allows you to look at the distribution and how the two are distributed throughout the cell cytoplasm. Question number 3. Great webinar, beautiful cells and impressive analysis. However, I work in a big pharma company, I have 0 coding skills, and I would need to get those graphs out as fast as possibly and preferably automatically. Is this something you can offer? But firstly, thank you very much for the compliment. It's always nice to hear that you enjoy the webinar. Secondly, work in a big pharma. Don't worry, I have zero coding skills as well, and I always find it easy to work with our data. And is this something that we can offer at the moment? So I cannot say too much on that last point at the moment, but hopefully soon we will have big news announcement coming from Nanolive. So all I can say is to keep checking our website over the next coming weeks. And also, if you haven't done it yet, subscribe to our newsletter so that when we do have news announcements of this type of nature, then you will be the first to know. Okay, so without further ado, that's all we have to do, all we have time for today. So thank you very much for joining me, and I hope to see you next time. Goodbye.